So the lecture today is on chapters 3 and 4 from the textbook. Still is introductory material, although I've tried to add a little bit to the lecture slide series to supplement what's in the textbook, especially what will come in the next lecture on the hypothalamus. So this lecture will include, as I go to the computer here, the pituitary portal system, the unique vasculature of this extravagant neuroendocrine structure. We'll look at the pituitary gland, both the anterior and the posterior pituitary, get into some details of that. And then we'll talk in general about the neuroendocrine axes, and the subsequent lecture will cover the hypothalamus, the hypothalamic hormones that control the anterior pituitary, and when talking about the pituitary gland, especially the posterior pituitary, we necessarily talk about the hypothalamic component of that anyway because it's a direct connection. But mostly in the second portion, we'll talk about the hypothalamic hormones that control the anterior pituitary, therefore. So then the hypothalamo-pituitary system is a neurovascular system. Any endocrine system, as I've stated repeatedly and continue to emphasize and reinforce, must be highly vascularized. You have to have a rich capillary bed, and we'll see that indeed throughout the base of the hypothalamus, called the mediobasal hypothalamus, and the pituitary stalk and the pituitary itself collectively, there's a rich vascular bed. Because we are discussing neuroendocrinology, we know that neuronal signaling is going to be very important in altering the release rate from the neuroendocrine cells of the hypothalamus themselves. So we know that we're going to look at synapses somewhere throughout. So the neuronal or interneuronal, such as an axodendritic synapse, is a prime example. And it's presented here today because it will come into our discussion a little bit to introduce the idea of regulation of neuroendocrine secretions. There's also neuroeffector types, such as a neuromuscular junction. This type of synapse we won't see much in this course, except most importantly for the innervation of the adrenal medulla, where we'll take a look at the acetylcholine as the neurotransmitter there, and as mediated, its effects as mediated through the nicotinic cholinergic receptor on the adrenal medullary cells, bringing about the exocytotic release of adrenaline. So we will see one example of that at least. Today, really, we're looking at the neurohemal junction. So these are neuron terminals, axon terminals ending on the extracellular space surrounding capillaries in the various portions of this neurovascular system that we will discuss. So then a picture that we should all be familiar with, the pituitary gland at the base of the brain, wherein lies the hypothalamus, and they are interconnected. The posterior lobe is the neural lobe. It is directly connected, and we'll see that there's actually axons coming from the hypothalamic neurons right into this area. I presented more so to show you the related structures. First, we use this structure, other than knowing that this is the anterior pituitary, we use this structure, the optic tract, the optic chiasm that has the optic nerves coming from the eyes and the optic tracts that split off to go back to the midbrain. The optic chiasm tells us that that is the anterior portion, or rostral, toward the nose. And then this large elevation here is part of the mammillary body system with the mammillary nuclei contained within it. The surface structure is the mammillary body. And that tells us that that is the posterior or caudal portion of the hypothalamus. So therefore we see there's an extension of the hypothalamus into the posterior lobe, and it joins with the glandular component, the anterior pituitary gland, and it is encased in this dense connective tissue capsule, and there's venous structures on either side, not shown here, from which the venules drain from the pituitary gland into the systemic venous system. And this is all in association with this structure called the sella turcica, or Turkish saddle, a deep indentation with the hypophyseal fossa in it within the sphenoid bone. So we're going to look at the development of the pituitary gland in a while, and this image is important to instill in our mind as we go through this. So the pituitary gland is encased in bone, except where there's a dense connective tissue or dura mater covering that surrounds it and is tightly adjacent all the way around the circumference of the pituitary stalk. Although the textbook doesn't go immediately to the vascular system, I decided to do that because it will provide us the details of this important vascular system and then begin to layer on some of the information about its role in the extracellular fluid communication between many of these neural systems and endocrine systems. So it's referred to as the hypothalamo-hypophyseal portal system. So the hypophysis, I guess the physis must be the hypothalamus, and below it is the pituitary gland, the hypophysis. So we're talking about a vascular system that carries, portal in French, the verb portage is to carry. A porter carries your baggage to the hotel room. So this PORT is telling us that it is a vessel system to carry something from one location to another. Obviously from some portion of the hypothalamus to the pituitary gland. So it's a set of vessels that interconnects two capillary beds, is the specific anatomical definition of a portal system. For example, the hepatic portal system, the first capillary bed is in the intestinal wall where the arterioles break up into the capillary bed. Venules, those capillaries coalesce into venules, ultimately veins, drain into the liver through the hepatic portal system, and then drain from the liver through a capillary bed in the liver, the second capillary bed, drain into the systemic venous system. So we'll see that a similar type of two capillary bed system connected by portal vessels exists in the base of the hypothalamus. And this is a very detailed illustration of the vasculature of this area. And I thought it was important to see this because typically we see it very simplified, as we will see in subsequent photographs. So I wanted to take you through this first by pointing out the optic tract here. So this is anterior, so now the nose is to the right. We've switched it around, so the anterior pituitary is now on our right-hand side. And then the mammillary body is seen here. This would be the left mammillary body. It is paired. There's a right mammillary body. Would identify the posterior limits of the hypothalamus for us. So there's vasculature in this area. Some of you might be familiar with the circle of Willis that's in this area, and branches come off the circle of Willis arteries to form the branches that go into the hypothalamus. The hypothalamus itself is highly vascularized, not only because of its neuroendocrine function, it's more so that it has chemosensory functions, and it's very metabolically active, so it needs to detect molecules in the blood for other functions that it has, and also has a demand for a good vascularization. It still doesn't quite compare with the capillary vascularization, very rich capillary bed throughout the base of the hypothalamus, where this collection of capillaries forms an elevation. This is called the median eminence. So MEDIAN means midline, like the median in a highway is the little grass that goes down the middle between two opposing direction highways. So the median eminence, and eminence, of course, is a protuberance. This is the median eminence here. The neural lobe is here, and it is connected through the pituitary stalk extending up through here. So that first image with none of this there, we reverse it in our mind to imagine this. So there is a superior hypophyseal artery and an inferior hypophyseal artery down here below that is providing the arterial supply to the mediabasal hypothalamus, anterior pituitary, and posterior pituitary, respectively. So we can follow the branches of the superior hypophyseal artery. There's an anterior branch here and a posterior branch here, and they give off branches that turn into arterioles and then feed into a capillary bed. And this capillary bed is made up of these elegant capillary loops. You can see that it loops up. There's little branchings of those, and it's on these loops that the neuroendocrine cells, in this case controlling the anterior pituitary gland, form their neurohemal junctions. These rich capillary beds coalesce into these portal vessels. There's a short one here. Excuse me, one of the long ones is here, the other long one is here, and then we see there's a branch of the posterior hypophyseal artery that goes up into the pituitary stalk just before the neural lobe and contributes to a capillary bed that immediately has the loops and immediately forms these short portal vessels that come down in, and the long portal vessels and the short portal vessels contribute to the capillary bed within the anterior pituitary. That capillary bed coalesces into the venules that ultimately exit and become the efferent veins of the anterior pituitary and goes into that dural venous sinus, which is basically the systemic venous circulation returned to the heart. So let's take another look at this from the more simplistic view. This capillary bed here in the median eminence is referred to as the primary capillary bed, and is where the neurons controlling the anterior pituitary form their neurohemal junctions. They would release their neurohormones into the interstitial space. They will diffuse down their concentration gradient into the plasma, be washed away down the long portal vessels, hypophyseal portal vessels, and into the capillary bed of the anterior pituitary gland. They would diffuse down their concentration gradient out of the plasma into the interstitial fluid, bounce around randomly, and eventually bump into the receptor and cause the response in the target cells. And we'll reemphasize that little molecular level trip all the time. So what I challenge you to do is imagine yourself in a saddle sitting on these molecules and following them wherever they go. So this capillary bed in the anterior pituitary is called the secondary plexus. So there's a primary plexus. The portal vessels, hypophyseal portal vessels, the long and the short, connect the primary plexus with the secondary plexus that is drained into systemic venous circulation. For the control of the anterior pituitary gland, we can simplify it and have a 1, 2, 3, 4, 5 stepwise fashion to now insert the neurons that are the neuroendocrine cells or hypophysiotrophic cells. They are neurons that are capable of causing some change. Trophism is change, or actually growth, but we'll just leave it as change in the anterior pituitary cells. So these hypophysiotrophic cell bodies of the hypothalamus have different secretory substances within them, and we'll go through a list of those. They're either releasing or inhibiting hormones that they dump into the interstitial space when they have action potentials and so on and so forth to stimulate the exocytotic release. Interstitial space at the primary plexus and it's picked up, the hormones are picked up by a capillary bed at the base of the hypothalamus, that primary plexus, are carried by the long and the short hypophyseal portal vessels to the secondary capillary plexus, so the bloodstream delivers hormones to a second capillary bed in the anterior lobe of the pituitary. The molecules of the releasing and inhibiting hormones diffuse out of the capillaries and act on the endocrine cells in the anterior lobe. So they diffuse out of the vessel, down their concentration gradient, into the interstitial fluid. They're bouncing around, Brownian motion and bulk flow through the interstitial space in the anterior pituitary. If they happen to bump into their cognate receptor on their target cells, they'll produce the response that would be expected, which is the hormones secreted from the anterior lobe cells enter small blood vessels that lead to the general circulation. So let's back up and saddle up that hormone now being released through exocytosis from the anterior pituitary cell. We're in the secretory granule. We fuse to the membrane. We enter the interstitial space, create a higher concentration of ourselves in the interstitial space of the pituitary gland, diffuse into the plasma down our concentration gradient. We are washed away into the systemic circulation. So as we'll see, these hypothalamic hormones are in high concentrations in this portal plasma, but once they wash out of the pituitary, if they're not destroyed there enzymatically, and get into the systemic circulation, they produce very low, sometimes even undetectable levels in the peripheral circulation. But they're high concentrations by the time they get to the anterior pituitary, and that's all we need. For the posterior pituitary, we can use this colorful illustration. So there are the hypothalamic, there are the neurons within the hypothalamus up here, all these different colors. The green and the pink, I guess, and the yellow are terminating on the primary portal plexus. So this one is showing us the nose is to the right, so it's the same orientation we had seen in a previous slide. And each of the hypothalamic hormones target the same colored cell in the anterior pituitary, so that would be the cognate target cell and the cognate receptor being on it. So they terminate on the primary plexus, the portal vessels carry it to the capillary bed in the anterior pituitary, and so on and so forth, as we had discussed. Posterior pituitary, we know that the axons of those neurons extend right through the the pituitary stalk and terminate on the capillaries within the posterior pituitary. So we'll find that these are larger neurons. They have to have a greater biosynthetic capacity. They have more rough ER, more Golgi, more vesicles, just more cytoplasm. They're called magnocellular neurons. These are smaller neurons because they only have to produce a high concentration in the hypothalamo-pituitary or hypophyseal portal system. So they're smaller neurons, less biosynthetic capabilities for secretion. So they're smaller, therefore called parvi or parvocellular neurons. And we'll emphasize that, re-emphasize it as we proceed. So this tract going through the pituitary stalk is called the hypothalamo-neurohypophyseal tract. In the central nervous system, a bundle of axons is called a tract, and we find these bundle axons passing through the pituitary stalk to form the neurohemal junction in the posterior pituitary. So it is the hypothalamic neurohypophyseal tract that we find in the pituitary stalk. We can take a look at another illustration. Again, they seem to get simplified endlessly. And what I'll do is point out a few things and then read through the legend on this, and then proceed to the next slide, which makes a point on this number 1 here. So we are seeing the superior hypophyseal artery forming the primary portal plexus in the median eminence. That capillary plexus coalesces into the long portal vessels here. The inferior hypophyseal artery gives a branch into the pituitary stalk that forms another component of the primary portal plexus. that coalesces into the short portal vessels that deliver the neurohormones to the anterior pituitary as well. And there they get out of the vasculature, target their cognate cells, the target cells with cognate receptors, and cause the release of the anterior pituitary hormones that's washed away into the venous, systemic venous circulation. The neurohypophyseal neurons that release in the neurohypophysis send their axons right through the pituitary stalk all the way down into the posterior pituitary to form the neurohemal junction. So the functional elements of control of the pituitary by the hypothalamus is what is being illustrated here. The peptidergic neurons that produce oxytocin and vasopressin, that's represented by 5 and 4. By 5, sorry about that, release their secretions into the general circulation by the posterior pituitary. So they are responsible for producing physiologically relevant levels of their own hormones in the systemic circulation. So if you draw venous blood, you can detect certain levels of oxytocin and vasopressin, or antidiuretic hormone is another name for vasopressin. So these neurons are big, magnocellular neurons, producing tremendous amounts, just constantly expressing the genes for oxytocin and vasopressin. They're separate populations, but it's shown just collectively here in this generic illustration. Post-translational processing of those hormones, packaging them, shipping them down, and storing them in large dilated axon terminals in the posterior pituitary. So there is a storage of these hormones in the posterior pituitary for release. The two general types of neurons are involved in the anterior pituitary regulation, the peptidergic neurons, 3 and 4, shown here. Form the releasing hormones, or it could be inhibiting hormones that enter the capillary plexus of the hypophyseal portal vessels. The second type of neuron is the link between the rest of the brain and the peptidergic neuron. These neurons, some of which are monoaminergic, are believed to end on the cell body of the peptidergic neuron in a conventional manner. We now know that this is true. This is quite an old illustration from the 70s. These actual synapses between monoaminergic neurons and the neuroendocrine cells has been demonstrated with very elegant methods at both the light and electron microscopic levels. So it's this neuron 1 that links the nervous system to the neuroendocrine cells that we'll talk about in the next slide. The other option is that these neurons with more traditional neurotransmitters in them end on the axon terminals of the peptidergic neurons in the median eminence. So there can be presynaptic axoaxonic types of interactions right in the median eminence. It is a highly organized vascular and endocrine structure. There's a tremendous amount of interaction between the supportive cells in this area, modified ependymal cells, and the axons that are there. So this neuron number 1 is a typical type of neuron we think about, with a neuroneuronal or interneuronal synaptic type of junction, one neuron to another. So neuron 1 to neuron 3, it's simplified. Every one of these neurons shown here is receiving tens of thousands of synaptic inputs, so it's very representative and simplified. That neuronal input takes us right to chapter 4, the point made in 4.3, neuroendocrine transducer concept. And this really is significant in thinking about the entire organism that we've talked about all along. You've got the nervous system, the somatic and the visceral, the conscious and the unconscious components that are linked so that we get coordinated motor and intestinal or visceral, including heart rate types of things, responses to internal and external environmental stimuli to the organism, and as well the third leg, the coordinated endocrine response. So we've got to link the nervous system with the endocrine system. So the hypothalamic neurosecretory cells are stimulated by neurotransmitters through the neurocrine type of a system, the interneuronal synapses that we illustrated. The excitability of these neuroendocrine cells, or these neurosecretory cells in the hypothalamus, has changed in that the firing rate will either be increased or decreased. Firing rate is in hertz, which is action potentials per second. Each action potential, as we should all remember from basic physiology, will bring about a certain quantal release of the hormones or neurotransmitter. Work the same way. They're related to the exocytotic release at the terminal, and as well, this type of input should increase the synthesis to replace what we just released. So we said release is going to always be coupled with increased gene expression to replace through synthetic mechanisms what has been released. There's always a basal synthesis rate, and especially when we think of the magnocellular neurons, they're constantly producing these hormones to keep a releasable pool in the posterior pituitary. And so if their firing rate is increased, say for vasopressin or antidiuretic hormone as it's called, if there's a sudden, you stay out all day and you don't drink any water and you become dehydrated, their firing rate is going to increase. So you're going to begin to deplete your stored granules that contain the ADH in the posterior pituitary. So you've got to not only replace the basal amount that was released, you've got to replace the store that's been depleted, and you've got to build that store back up and sustain it as long as you've got the increased activity. So these neurons are going to increase their synthetic activity to replace what was released on the basal level, build the stores, and then fight to keep the stores and the basal amounts supplied throughout the duration of the dehydration. These systems can be exhausted, of course, but that's what these cells will attempt to do under the appropriate stimuli. The hypothalamic hypophysiotrophic neurons therefore convert neural information into hormonal output. And this is what makes them indeed a true transducer, neural signal to endocrine signal. And this is then representing the responsibility of this transducer system for bringing the endocrine system under the control of the nervous system. So psychogenic factors, just emotions, can activate neurons within our nervous system without any external or internal environmental sensory input, and can end up activating neurons that will dump excitatory transmitters onto the neuroendocrine cells in the hypothalamus. Those neuroendocrine cells increase their firing rate and increase the release of the hormone. So the neuroendocrine cells, those neurons in the hypothalamus, are the transducers that transduce the information of the nervous system into a humoral mediated or extracellular fluid mediated signaling system, the endocrine system. So the authors provide this figure, 4.2, to make this point, and I've talked about it already. So we're seeing the neuroendocrine cells illustrated from the hypothalamus. So there's dendrites and the cell body, the soma. So you can have axodendritic synapses, axosomatic synapses to make this a neuroendocrine transducer cell where the neurotransmitters are regulating its activity. You have excitatory transmitters, inhibitory transmitters working together to produce baseline, and then bringing information in to change above or below baseline the activity of this neuron. That activity is the firing rate, the number of action potentials that it generates per second, or the hertz firing rate of this neuron. And we'll talk about neural integration relative to neurotransmitters and neuropeptides throughout the course. So this neuro signaling through the neurocrine interneuronal synapse system is an integral part of this course. So all of that impacts the action potential, which we know when it arrives at the terminal will trigger calcium influx, typical type of exocytosis for hormonal release. At the same time, this neuron has received that input. It doesn't really know that it's released anything, because if you could block release, you still would have the same signaling in this neuron to tell the nucleus to increase the expression of the gene for the biosynthesis of the hormone that should be coupled to the firing rate. And as the synthesis builds, then axonal transport, anterograde axonal transport will increase such that you can restore the storage component of this. So we don't get synthesis and immediate transport to the site of release and the release. There is a coupling between synthesis and release, but there is a temporal delay in transcription, translation, post-translational processing, the packaging, and the transport, and the preparedness as a readily releasable pool here in the terminal. So the release, we know, is into the circulation for the neurohormones, or into the synapse if it's going to be a neuropeptide or even a neurotransmitter somewhere within the nervous system. So we have the cell body, the axon, and the terminal as the key components, if we're considering this as a hypothalamic neuroendocrine cell, key components of the neuroendocrine transducer. Well, now we can talk about the pituitary gland in general, and this will bring us to the end of this part of the lecture, so we'll work our way through, and hopefully we can complete the component on the anterior pituitary. If not, we'll include that in the next lecture when we discuss the hypothalamus, which really will be a discussion of control of the anterior pituitary. So it is divided into three main parts. There's an anterior lobe, as we've been seeing, called the pars distalis. It's the most distant from the hypothalamus. There is an intermediate lobe called the pars intermedia, and a posterior lobe called the pars nervosa, and another one, as we'll see, called the pars tuberalis. But we can live with just this terminology for three of them. The adenohypophysis is a term that includes 1 and 2. Adeno means glandular, so this is the typical peripheral type of gland that we would think of as the anterior pituitary gland is. So the adenohypophysis is 1 and 2 together, the anterior lobe, pars distalis, and the intermediate lobe, the pars intermedia. And the neurohypophysis is the posterior lobe, the pars nervosa. Let's take a look at the development of the anterior pituitary gland. Just checking, since some of the slides are not fitting onto the screen, but that's all right. You'll have the printout to go by relative to the streaming video. So here in this illustration, we have panel A, B, and C. Panel A is a low magnification showing the human fetus, the development of the forebrain here in blue. The mesoderm would be the red, and the ectodermal layer would be just on the surface. And we can see that the oral cavity is forming here. So in this case, the nose is primordium of the nose would be to the left. And so this is the oral cavity here. The roof of the oral cavity, actually the pharynx, and just at the end of the primordial oral cavity, the pharynx here, the roof of that pharynx will begin to invaginate, as well as the floor of the diencephalon will produce a solid core of cells that begin to push into the underlying mesoderm. These two structures we see are called the hypophyseal pouch and the neurohypophyseal bud. And this is at about 36 days, so basically in the first month of gestation, just shortly after the first month of gestation. They continue to grow, so panel C, let's see if we can find it here. Well, 8 weeks is going to be D, but so somewhere in here, 36 days is about 5 weeks, so 6th and 7th week, they approximate each other and make contact, so I'm sure there's a lot of intracellular signaling here to bring about this type of development. We see that a duct actually forms into the cavity within the hypophyseal pouch, and the hypophyseal pouch is differentiated from the oral ectoderm, the surface layer of cells on the oral cavity. So most glands are generated from epithelial cells as an origin. So the neurohypophyseal bud and the hypophyseal pouch make contact with each other. There is an indentation at this primordium of the floor of the third ventricle. We see this large ventricular system in the early developing nervous system that we know persists. So the floor of the hypothalamus is another term we can state here. And this becomes funnel-shaped, so it's called the infundibulum. The infundibulum is also the term used for the pituitary stalk. It's overall funnel-shaped. So 8 weeks is shown here in D as we proceed from 8 weeks to much later. Even the adult appearance is apparent here, but this is still in utero fetal stage. We see that there's a regression of the stalk of the hypophyseal pouch. This is also called Rathke's pouch. We'll see that on another slide in a moment. So it really is a cyst-like structure or vesicle-like structure that is going to contribute to the formation of the anterior lobe of the pituitary. We see the pars intermedia is that portion that becomes adjacent to the anteriormost surface of the primordium of the neural hypophysis, the neural lobe, and the little tail extends up toward the pituitary stalk at the base of the hypothalamus, and this ends up producing these structures. Notice the developing sphenoid bone here that will ossify and surround this, forming the sella turcica with this depression, the hypophyseal fossa. We see the vasculature forming all around the pituitary within the layer of the dura mater. The arteries and veins would already have developed themselves in here. They're just not illustrated. The bone becomes ossified. These little remnants of the hypophyseal stalk from the pouch are supposed to disappear completely. So whenever you see something like this remaining, this means that you could have an ectopic pituitary tumor developed from these, but we're not into pathology here. We're just assuming that these will all go away, and the adenohypophysis is present here. The opening, the cavity of the hypophyseal pouch or Rathke's pouch does disappear, except for these little nodular type of colloid containing vesicles, and that persists into adulthood, as we will see. And we end up with a pars tuberalis surrounding the tuberal portion of the infundibular stalk. So funnel shape does have a little tubular portion on it, if you picture a funnel. So that's the part of the pituitary, anterior pituitary that surrounds that is called the pars tuberalis. I don't think this should be the tuberalis here. This should be pars distalis. We should be able to correct that from our own terminology now. Pars distalis here, and the pars intermedia most closely adjacent to the neurohypophysis. If we make a coronal section through this stage of the human embryo, at about the 7th or 8th week. We can see at the base of the brain, the forebrain, these structures that we've been talking about. First, we'll get our orientation. This is the developing cortex up here on the right and the left side being separated. We know the corpus callosum will develop to connect those. We see choroid plexus pushing into these two fluid-filled cavities, the right and the left lateral ventricles that drain through the interventricular foramen into the third ventricle that has the thalamus and the hypothalamus as its walls. So here we see it's called the hypothalamic sulcus. It separates the thalamus from the hypothalamus, and we see the third ventricle is the fluid-filled space between the right and the left hypothalami on each side. Right and left hypothalami converge at the floor, where the median eminence is developing at the attachment of the infundibular stalk. And then we see the neurohypophyseal bud here in the center, and on each side of it, the hypophyseal pouch or Rathke's pouch. So the roof of the oral cavity, the pharyngeal cavity, would be down here below. This slide summarizes it then. Now we have the nose to the right, so anterior is to the right. So we see the mouth cavity or the oral cavity, the roof of that pushes up. The base of the forebrain at the diencephalon where the hypothalamus is pushes down, produces the infundibular process and this funnel-shaped depression or fossa. They join together, create that association with a loss of the connection with the epithelium lining the pharynx. The cavity is still there, but it disappears as the number of cells grow. Differentially in the pars distalis, a little bit more, the number of cells increase in the pars tuberalis, but the pars intermedia does not experience an extensive cell growth, therefore becomes very small. Ultimately, as the bone ossifies, the pars distalis grows, the cavity is closed off until finally we see this adult appearing structure with the pars tuberalis surrounding the infundibular or pituitary stalk. We see the mammillary bodies there for orientation. And the neural lobe extending posteriorly here. If we do a cross-section of this, we see that the cleft of the Rathke's pouch is still there, but it will disappear by birth, definitely. With this being the pars distalis or pars glandularis, pars tuberalis up there, and the pars intermedia, and then the pars nervosa. So now we can begin to talk about it as an adult structure as we've gained an appreciation for its development. First with the neurohypophysis. So the terminal extension of the pituitary stalk, as we saw, it is referred to as the infundibulum, the funnel-shaped portion closest to the hypothalamus. The pituitary or infundibular stalk contains the hypothalamo-hypophyseal tract, as we said, a collection of axons within the CNS is a tract. So it extends right through the base of the hypothalamus, through the stalk, and into the neurohypophysis, as we had said. So we can use this colorful illustration to see that for two nuclei, the paraventricular nucleus. First, we see the optic chiasm, so now anterior is to our left. The mammillary bodies would be back here to the right for posterior. The paraventricular nucleus is in the wall of the hypothalamus, lining the third ventricle, so close to it, parallel to it, not immediately adjacent to it. There's a periventricular area, as we will see, that's closer to the actual surface of the third ventricle. This is the paraventricular nucleus. We see a rich arterial supply, and the supraoptic nucleus above the optic tract, that is the extension of the optic chiasm where portions of the optic nerve cross. So above the optic system is the supraoptic nucleus. We see blue neurons in each location whose axons pass straight through the infundibular stalk in to terminate on the capillary bed 3 in the posterior pituitary. The two hormones of the posterior pituitary are vasopressin or antidiuretic hormone and oxytocin. If we take a look at these octapeptides, 1, 2, 3, 4, 5, 6, 7, 8, this glycine and the amidation are post-translational processing that we'll talk about at a later time to stabilize the molecule. We also see disulfide bonds in here that obviously are important for forming the biologically active structure of these peptides. So there's a lot of post-translational processing that can occur with these. So these are 8 residues, so they are octapeptides. We know that the vasopressin stimulates water absorption by the kidney. We'll look at these in detail in other lectures. And the oxytocin stimulates uterine contractions and milk ejection by targeting different cells and different target organs and tissues. So we see that there's quite a homology in these. The third residue is phenylalanine in vasopressin, isoleucine in oxytocin, and then the 8th residue is arginine in vasopressin and leucine in oxytocin. So not a tremendous difference. So that would be this isoleucine here and this leucine here in oxytocin that differ between it and vasopressin, and that is enough. And they both have the disulfide bond, so their overall structure would be the same. Those two amino acid residue differences make a difference in the binding to the receptors in the target cells. The antidiuretic hormone to the renal tubules in the kidney, and the oxytocin to specialized cells in the breast tissue and the smooth muscle cells in the wall of the uterus. So we'll follow through with this as an example of target cells in looking at the glandular portion, functional portion of the breast tissue. We note that they're the glandular portions, the secretory portions, with the ducts that we know have ampullae and they terminate on the nipple. So mechanical stimulus to the nipple, of course, is going to cause the secretion of oxytocin. When the oxytocin is released into the systemic venous circulation, back to the heart, through the lungs, back to the left heart, through the aorta, from the branches of the aorta, finally into the breast tissue, into a capillary somewhere nearby here, diffuse out, bounce around in the interstitial space, and you have 2 target cells within this target tissue of the target organ. These are the secretory cells, the milk-producing cells, and they are surrounded, they form this cup-shaped structure called an acinus, A-C-I-N-U-S, or acini, A-C-I-N-I, right, for plural. This acinus is showing the cavity inside and cells on the structure that are modified epithelial cells that behave like they have contractile properties, so they're called myoepithelial cells. They're not really smooth muscle cells, but they have contractile capabilities. So you're the oxytocin, you're riding the oxytocin, bouncing around, you bind to your receptor on this cell, you bump into the other cells, but they don't have the receptor, so you can't do anything to them. Bind to the receptor on this cell, and cause intracellular changes that cause it to contract. It'll empty the contents into the duct. At the same time, the smooth muscle cells are targeted, bouncing around randomly, bind into the oxytocin receptors on the smooth muscle cells in the duct, and the milk is ejected at the nipple, and that is the milk letdown reflex. So this slide begins a discussion of the adenohypophysis, and we'll begin that. We'll cover that in the next lecture along with the hypothalamus and the hypothalamic hormones. So I appreciate your time and attention today, and we'll see you next time.